Today we've reached the last topic in Chapter 5, and this is the topic that we've been building towards throughout the whole chapter. So I mentioned that I do the topics out of order. Most textbooks start talking about the visual system at the very beginning, but I've chosen to end with the visual system now that we've built up all the correct terminology and to have a better understanding of some of the topics that are going to help us grasp what ends up being one of the more complex of our sensory systems. So, as per usual with our systems, we start off with what is our stimulus. And for the visual system, our stimulus is light. And when we get technical about it, light is a form of electromagnetic radiation that travels as a wave. If you're not super familiar with electromagnetic radiation, then you might not realize that our visible spectrum, the light that we can see, makes up only a very small portion of all of the different types of energy, or the things that we can also categorize as electromagnetic radiation. So just a little bit below our the end of our visible spectrum here. So at that 400 nanometers end of our visible spectrum, if we go below that, we have UV, X-rays. If we go above the upper limit here of our 700 nanometers for our visible spectrum, we have infrared and microwave and so on. So this just tells us that light is a small window of all of the possible electromagnetic spectrum. And in fact, other species have the ability to see into either the UV range or the infrared range, as well as in that visible light range. So if we think of birds, most birds anyways, can see in the UV range. Some reptiles can see into the infrared range. So it just means that their sensory receptors are sensitive to a different range or spectrum of this electromagnetic radiation. But for us and with human eyes, we can see between 400 nanometers and 700 nanometers, and that wavelength is going to correspond to what color we perceive light to be. And so this should start sounding a little bit familiar because we've already talked quite a bit about the properties of waves when we were looking at sound and sound waves in our section on our auditory system. And so here we're going to see a lot of parallels where amplitude in our waves, so if we were looking at sort of one of those nice curves, amplitude being the deviation from that center line. So a larger amplitude, if we had a much larger wave, that just has more energy. And when we were talking about sound, larger amplitude meant louder sound. But for light, larger amplitude is going to be a brighter light. So our amplitude relates to brightness. Our wavelength, which was, again, I can put my little squiggle back, is that distance between adjacent peaks. Actually, let's see if I can draw one that's just a little bit smaller so we can actually get a cycle here. So between the adjacent peaks here, that would be our wavelength. So that is our perception of color. So as we had pointed out on the last slide, our visible spectrum ranges from that 400 nanometers, that's our wavelength, up to 700 nanometers. So the wavelength is going to correspond to what color we perceive it as. And then we can also talk about purity, where in our sound waves, that was talking about the quality of the tone. And for light, it's just the quality of the color that we're seeing. So if we have a very pure signal, a very pure wave, then we're going to have very bright, intense colors. So saturation will be high. If we have a messy wave, a mix of wavelengths where it's impure, we would have a very low saturation. So that's how those categories all relate. Next, we're going to start talking about the important structures in the eye that are involved in our ability to see. And so you're going to find that I'm going to stick on the slides that have pictures, but I'm also going to be talking about the information that's on the slides with words adjacent to them. And I find because the sort of images and the words tend to go nicely together, I just want to let you know that that's what I'm doing. So even though I'm going to be on this screen, we're going to be talking about this information, and I'm going to keep doing that as we move throughout the next couple of slides, just so that you're oriented with what's going on here. But when we're looking at this image of the eye, we are sort of envisioning what's happening as light enters the eye. So this would be our light. And so we're going to talk about structures in the order that they would pass through. So when light is first entering the eye, the first thing it's going to enter is the cornea. So it enters through the cornea, which is that clear outer surface of our eye. And it marks the boundary of the anterior chamber. That's where our aqueous humor is. So that's a fluid that just helps that part of our eye hold its shape. And so we're going to be passing through something that is technically marked on here, and this is our pupil. So our pupil is the gap that's created by our iris. So the pupil is just the black hole in our eyes. And the reason why it's black is because it's letting us see onto the inside of our eye, where we don't actually have as much light as we have on the outside. So it appears black just because of a lack of light. And so the pupil isn't technically a structure, it's just a hole that's created by the iris. And the iris is a colored ring of muscle that can contract or sort of expand in order to allow more or less light into the eye. So the size of our pupil is dependent upon what our iris is doing and how much light it is allowing into our eye. So that's the first step there. The next thing we do is we have light pass through our lens. And our lens is kind of just what it sounds like. It is something that is going to bend and shape the light that comes into our eye. So our lens can actually make adjustments, and these adjustments are called accommodations. And that means that our lens can either sort of round itself out, get squishier in the middle, so it can get fatter and rounder when we have an object up close, when we're looking at something near to the eye, or our lens can sort of flatten out and get taller and thinner when we're looking at something that's far away. And those expansions and contractions are controlled by the suspensory ligaments, so they control what our lens is doing. And then when the light is passing through this lens, it's going to sort of get shaped and bounced to face on the back of our eye. So my lines are actually probably a little bit crooked here because we would really like to focus on our fovea. The fovea is our focal point. So it should be focusing the light in and around our fovea. And you'll notice that when the light crosses like this, this is basically showing us that we see everything inverted. So if we had an arrow, if we were looking at, let me get rid of my light here. If we were looking at an arrow in front of our eye, we would see it actually inverted, so it would project onto the back of our eye upside down. So this line crosses over and moves up here, and this line crosses over and moves down there. That's just one of the properties that our lens does. So when light hits the back of our eye, when it hits our retina, almost like the projection screen that runs along the back of our eyes, we're seeing it inverted, and our brain just corrects for that inversion, which is pretty cool. But as that inversion is happening, we're passing through our vitreous chamber, and that's full of vitreous humor, which is a liquid, sort of a gel substance, and that's what gets our eyes to hold their shape. And so we mentioned that our fovea is our focal point. This is where we get the most detail from our vision. So that's where, when we're looking at something up close, when we're paying attention to something, that center point that we're focusing on is going to fall on the fovea. It's going to fall on a point that gives us the most detail possible. And it gives us the most detail because that's where our sensory receptors are most dense. And we'll talk a little bit more about our sensory receptors in just a moment. And I kind of also mentioned that our retina is this screen-like substance at the back. That's what all of our light is being projected onto. And at the level of the retina, that's where light is going to land. So the retina absorbs light. It does a little bit of processing where we have far too many sensory receptors in the retina enough for each one to send a neural signal back separately to the brain. So what happens is that we'll actually condense the message, where a couple of sensory receptors will be sort of averaged together, and that averaged signal will be sent along a neuron into the brain. But each individual receptor does not have its own designated pathway into the brain. They're going to share pathways. So that's why our retina does very low-level processing by averaging across a couple of sensors. And then that's where all of that information gets sent to the brain. And what we don't see here is that sort of from the sensory receptors in that retina, they're going to have neurons that come and pass out of the eye. So these neurons come and form our optic nerve, and that's going to head off to our brain. And where the optic nerve leaves the eye, there's sort of this gap here. And because that's where all of the neurons from all along the back of our eye are exiting, so it's a lot of stuff leaving the eye, we can't actually have any receptors there because all of our neurons are in the way. And so we technically have what's called a blind spot here. Or if we wanted to get technical, we could call it the optic disc. And that's just where that optic nerve leaves the eye. I mostly refer to it as a blind spot because that's what it is. If we don't have any receptors there, we don't have any perception there. We don't have any vision there. And you don't notice our lack of vision there because our brain actually fills in the blanks. So our brain will correct for the fact that we can't see there and just make some assumptions. So most of the time we don't even notice that there's a gap there. Up until this point, I've been talking fairly vaguely about our sensory receptors, but I haven't actually given them a technical name yet. And this is where we're going to start doing that. And the reason why I didn't give them a name up front is because we actually have two different kinds of sensory receptors. So we have rods and we have cones, and they're both specialized to function in very different ways. So our rods are really good at detecting sort of the presence or absence of light. So they'll give us a sort of understanding of our environment in terms of black and white. Is it bright or is it dark? And because it doesn't rely on anything more than just the presence or absence of light, the rods tend to focus very well in low light conditions. So when we're out in the middle of the night, we rely on our rods to operate and keep us seeing properly in those low light conditions. Our rods are really good about giving us information about movement, but they don't have a ton of information about detail, and they definitely don't tell us anything about color. That's why at night it's a lot harder to tell what color things are. It's because we're relying on our rods, and it's harder to tell things apart because they don't see that color. And so, of course, our other kind of receptor cells are cones, and cones detect color. So they're responsible for our daylight vision. They need a lot more energy in order for us to figure out what color we're seeing because we're perceiving different wavelengths of light. So that only works when there's a lot more light entering our eyes. And that's why we rely on our cones during the day, but our low energy requiring rods can function well at night. And our cones also give us a lot of information about detail. So detail and color come from our cones, and then just black and white and sort of movement information would come from our rods. And we go through a process where, okay, so if we use rods in low light conditions and cones in bright light conditions, how do we switch between the two? And this is the process of adaptation, where we can sort of adapt or change how we see to become more or less sensitive to light as needed. So imagine leaving a movie theater after you've been to see a matinee. So you went to the movies in the afternoon and you walk out of the theater at 2 o'clock and the sun is so bright. It was nice and dark inside the movie theater. You were able to use your rods with no problems because of the low light conditions, but you step outside and the sun is absolutely blinding. And that's because there's too much light for our rods to function effectively. So we have to switch over to using our cones. And that process takes quite a bit of time. Within the first minute to 2 minutes, we'll have a decent switchover where we can start functioning. So your eyes are going to hurt for the first few minutes and then you'll be okay. But it actually takes up to 20 minutes for a full switchover from rods to cones, or if we're going from cones to rods. So if you're going to bed at night and you turn off all the lights and then it's too dark for you to see where your bed is, that's sort of going in the other direction. And so that process of adaptation, of swapping between rods and cones, takes a little bit of time as we adjust or accommodate to what's changed in our environment. And on the next slide here, we're actually looking a little bit closer at our retinas. And here we can start seeing why rods and cones have the names that they do. Our rods are long and thin and kind of rod-shaped, and our cones have a tapered cone shape to them. So the rods are sort of skinnier and longer, and then the cones are tapered and cone-shaped. And what we're going to notice here is that we're looking at light entering the eye. This is our retina. So this whole part here is what we would call our retina. But you might notice something a little bit weird here. And what's going on is our receptors are our photoreceptors. Those are the ones that receive light. So our rods and cones, this part of those cells is what reacts with light to start sending a signal into our brain. But that's at the back of our retina. In order for light to get through to that point, we have to pass through all of these other cells, all of this support network that's in front of it. So light has to pass through our ganglion cells, our bipolar cells, and the front part of our photoreceptors in order to get to the point where they can actually activate those photoreceptors. So if we were trying to design a more efficient setup, we might actually reverse this and put the photoreceptors on the on the front side, so they'd intercept the light first. But instead, the light has to pass around all of these different kinds of cells to get where they need to be. And then once the light has interacted with these structures, with the rods or the cones, with that photoreceptor portion, then the signal gets sent back outward, back through, and it'll pass into our bipolar cells, the ganglion cells, and then out to the brain. So that'll go along the optic nerve and towards the brain. And you'll notice, or you might notice here, that we can see what I talked about earlier, that low-level processing is happening at this level of the bipolar cells. So here we have 6 different rods, and they're all being fed into a single bipolar cell. So this bipolar cell would be taking the responses or the firing from each of these rods and condensing it into one signal that gets sent from that bipolar cell to the ganglion cell and then down to the optic nerve. So this would be low-level processing where we're averaging out the responding of all of these rods to send that signal to the brain. So each individual rod doesn't have a designated bipolar cell. They're going to share that network. For our cones, those give us a lot more detail, and they're sort of more important for our color vision, and there's a lot of what we rely on for our detail. So our cones are special, and they end up getting a bipolar cell to themselves. There's a lot less of that condensing, so we're going to get a more precise image from an individual cone cell, which kind of makes sense when we look at those functions there. And before we talk about that pathway, I'd actually like to talk about the distribution of our rods and cones in our eyes. So this is a little bit of a weird graph. So let's orient ourselves first by looking at this image of the eye. So this is a top-down view of our eye. So this would be the front of the eye, this would be the back, and then this would be sort of where our nose is, and that is on the outside, sort of towards our ears. And so here we've marked our fovea, or our focal point, as 0 degrees. So that is our reference point. So we're measuring outward from our fovea. So as we go around the back of the eye, we're going to be reporting in degrees how far we've moved or pivoted from that fovea. So that's what we're recording as our axis here along the bottom. So our x-axis is how far away from the fovea we are, sort of out to the left or into the right. And the first thing we notice here is that our optic nerve leaves at about 10 degrees, a little bit closer to 20 degrees. And so we have a cutoff where there are no receptors there, as we talked about earlier. So it is a blind spot because that's where the optic nerve is leaving. There are no receptors there. So we have no signal, no rods, no cones in the blind spot, and that makes sense. Next, let's look at our red line. So that's where our cones are distributed. And so we've labeled it here as the peak of our cones, meaning the place where we have the most density of cones is right in the middle of our fovea. And as we move away from the fovea, as we wrap around the back of the eye, the number of cones drops off very dramatically, and we end up having quite a low concentration of cones outside of that fovea. But at the fovea, there are so many cones that they pretty much crowd out all the rods. And we'll notice then if we look at our blue line, which is our rod distribution or our rod density, there are no rods in the fovea. They've been crowded out by those cones. So what the rods do is they have the highest density that they can as close to the fovea as possible, but still outside of the fovea. So they end up being more present close to the fovea, but not at the fovea. And then they taper off in concentration as we wrap around, but they don't drop off as dramatically as our cones do. And this is because we have far more rod cells than we do cone cells, so the drop-off is a lot more gradual. So in the eye overall, we have lots more rods than cones. But if you were asked at the fovea, which kind of cell do you have more of? At the fovea, we have more cones than rods. That's a good visualization of the distribution of these two types of receptors. So next, we can talk very, very briefly about the visual pathway, and we could go into a ton more detail on this, but I find it's a lot less important for this class than it might be for, say, a sensation and perception course that you might take later on. So I'm going to gloss over some of the details here, and you're going to figure out very quickly that this is something that we've already talked about when we were looking at our split brain patients. So our visual pathway is going to depend on sort of what side of the eye are we seeing something on. So if something is on the left side of either our left eye or right eye, so that blue side, we're seeing that left side of our vision is going to transfer over to the right side of our brain. And that's the same as when we were looking at our sensory perception and our motor control, where we have that crossing over effect, where the right side of our brain is responsible for the left side of the body and vice versa. So with our eyes, the things that we see on the left side go to the right side of our brain. And these red things that we see on the right side go to the left side of our brain. And that crossover that is going to happen where sort of the right side of our vision and our right eye needs to cross over to the left side of our brain. And the left side of our vision on our left eye needs to cross over to the right side of our brain. So that cross point where the fibers cross over is called the optic chiasm. And that's going to be going across our corpus callosum. I almost forgot the word there for a second. So in our split brain patients, we were looking at what happened when this was severed. What happens when this part of our information can't get over here and this part of our information can't get over there. That's what we were looking at in that case. But this is the pathway that we're looking at here. So we have that crossover at the optic chiasm. Then we travel along that optic tract towards our occipital cortex. And then at the level of our occipital cortex, that's where we start having some processing of that visual information. And that's the part that I want to focus in just a little bit more. So we're going to talk about something really cool, which are called feature detectors. And these are neurons that are going to respond selectively to different kinds of features. So there are some feature detectors that respond to lines. So maybe we have neuron A, and it responds to vertical lines. And then maybe we have neuron B, and it responds to horizontal lines. And what that means is if there is a vertical line in our vision, then neuron A is going to fire or have action potentials telling us that there's a vertical line. And B would fire when we have horizontal lines present. So we get action potentials when it sees a horizontal line. And new technologies allow us to measure sort of individual or single neurons firing. So we could look at what happens when, okay, so maybe it isn't a perfectly vertical line. What if we rotate it a little bit and now it's maybe 5 degrees or 10 degrees off of the vertical? Does our cell still fire? Or does it fire less frequently? Is it telling us a less intensive signal? Or is it pretty much quiet at that point? So we can look at things like that, look at these feature detectors to determine what changes we can make that would make a feature detector detect something or not detect something based on those adjustments. And as we've done more and more research into this, we've found that we actually have even more complex versions of these feature detectors. So in humans and monkeys, we have cells that fire in response to seeing faces or even face-shaped objects. So if we saw an oval, we'll pretend that's an oval, and there are 2 circles on it at about eye height, those cells would start firing to say that we have something face-shaped that we're perceiving. And this might help explain why humans are so good at seeing faces, even if it isn't actually a face. So maybe you're looking at a knobbly potato, but it has some spots on it that look like a face, and so we automatically treat it like a face. Or maybe we're really good at seeing faces in clouds or water spots or whatever. That sort of pre-programming to look for faces comes from us actually having specialized cells that respond to those kinds of arrangements. So those can act as kind of shortcuts that tell us when we see different kinds of features that are really important to our sort of functioning and perception and survival. So it's really helpful to spot faces because that helps us recognize others of our kind who we might want to interact with. We're going to pivot briefly in terms of topics here, and we're going to start talking a little bit about color mixture. And the reason why I'm doing this is because we're going to start talking about the different theories on how we perceive color. And in order to do that, we need to talk about adding together different colors of light. But I find that a lot of students, they only have experience with color mixing in terms of, say, mixing paints together. So if you think of back in grade school and you did finger painting and you'd mix yellow paint and red paint and you'd get orange paint, that is what we would call subtractive color mixing. And this works because when we have items, when we have things that have color, say like red paint, we're having those specific items remove some of the wavelengths of light. So if we think about our visible spectrum, white light would be the presence of all of those visible wavelengths, everything between the 400 nanometers and the 700 nanometers. So when white light shines on red paint, that red paint is going to absorb all of the wavelengths except for red. So it subtracts everything except for red. And because it reflects red light, that red light will reach our eyes and we will see that paint as red. So when we're mixing 2 paints together, so if we have our yellow paint, and so that has absorbed all of the wavelengths except for those that relate to yellow, so the yellow light would reach our eyes. And we have red paint where it's absorbed everything except for the red wavelengths. When we mix those together, we're going to get a little bit of the red wavelengths and a little bit of the yellow wavelengths, but the paint is mostly going to be absorbing or subtracting most of those wavelengths of light. So there's less light than there was originally. And if I hop to this next slide, we have a visual of that, where they've mixed together these different colors, and when we have an overlap of all of the colors that we could possibly mix, we end up getting pretty close to black, the absence of light. because we have subtracted all of the wavelengths except yellow, and then all of the wavelengths except blue, and all of the wavelengths except red. And so we're left with no wavelengths left, and we end up with a dark color. In practice, when you've done this with, say, paint, you probably ended up with sort of a muddy grayish color, because it's really hard to have no reflectivity, but that's the theory of what this would look like. In contrast, when we're looking at our eyes, we're looking at different wavelengths of light coming in. So we're not looking at the process of subtracting out wavelengths and reflecting sort of specific types. Instead, we're looking at what's called additive color mixing. So we're focusing on adding together any incoming waves of light, those sort of different frequencies, those different wavelengths. Our eyes are going to be superimposing or adding those together. So we're putting more light into the mixture. So here on the left side, when we have green and blue and red, as we add those together, we're going to get closer and closer to white light. And just to remind you why this could happen, let's scroll all the way back to our visible spectrum diagram. And so we were looking at red and blue and green. And so red, blue, and green would be sort of the upper limit, pretty close to the lower limit, and then the middle of our visible spectrum. So by adding together sort of the high end and the low end and the middle of all of those possible wavelengths, we get a pretty decent approximation of the visible spectrum of all of those wavelengths. And all of those wavelengths are part of white light. So the more wavelengths we're adding together, the closer we're getting to that white light. Now, if that hasn't sunk in immediately for you, that is perfectly fine. This is a little bit of a strange concept. And so I always include this YouTube link at the bottom here. And this is just a video where someone actually shows you mixing together paints and mixing together lights to illustrate the difference between additive and subtractive color mixing. So if you'd like, you can pause this video and go watch that YouTube video to help clarify any sort of leftover confusion. But from here on out, we're going to be talking about additive color mixing, where we're adding together different wavelengths of light that are coming into our eyes. And so we're going to look at a couple of different theories using this additive color mixing that explain how we see the world around us in color. The first of three color perception theories that we're going to look at is the trichromatic theory. This was proposed by Young and Helmholtz in the mid-1800s, so this is quite an old theory. And as you'll find, some of the ideas for this theory work and have been carried forward, but it isn't necessarily the whole picture. So as we're working through all of the different theories, we're going to kind of be building on them and adding them together. But in this theory, they've noted that the human eye has three different types of receptors that have differing types of sensitivities to wavelengths of light. So what that's saying is that we have three different kinds of cones, and each of those types of cones will have a sensitivity to different wavelengths of light. So they're going to be sensitive to either red, green, or blue wavelengths of light. And the sort of comparative responding of those different kinds of cone cells can tell our brain how much red, green, and blue that we're seeing, and we can add it together in our brain using the additive mixing process. So if we have, actually I can scroll back here, so if we have red and green, we're going to perceive it as more yellow. If we have green and blue, we're going to see it as sort of a lighter blue. If we have blue and red, we're going to see sort of a pink color. And if we have all three of them firing very dramatically, then we have white. And so this is a very simplistic sort of image here, but we could get all sorts of different colors. That's how we could perceive all of the varieties of colors that we have in our surroundings. And if we look at this diagram here, where we have our sort of span of the visible spectrum, they've gone with the more precise here. So instead of just saying vaguely 400 to 700, they've expanded it up to 750 and down to 390. That's not overly important. It's all about where you choose to cut off. But what we care about from this diagram is that they show us the blue, green, and red, so short, medium, and long wavelengths. So we can see that each of these would have a curve. So this is saying that our short or blue cones would respond really, really well in the range of about 440 or so, and it would continue responding, but sort of less so, a little bit below and a little bit above. So we'd get the most responding at the peak here, and then it would taper off to the left and to the right. For our, let's pick a better color. So for our medium, for our greens, we'd respond really, really, really well around this sort of 540 range, and we would respond a little bit below and a little bit above. But that's where we'd see the most responding with this particular wavelength. And so if we had light that was coming in with this wavelength right here in this 480 range, we'd get some firing from our blue cones and we get some firing from our green cones, just about the same amount. And then we get a little bit of leftover firing also from the reds. And so our brain could interpret that this much firing in the blue cones and this much firing in the green cones and this little bit of firing in the red cones, that corresponds to this particular wavelength of light. So this is the color that we're seeing. So that's how these three different kinds of cones could explain how we perceive color using additive color theory. And this process does a great job of explaining color blindness. So many people who are colorblind are what are called dichromats, which basically means that they are insensitive to one of these wavelengths of light. So if you don't have the blue cones, then you wouldn't have any firing for this particular area. So we'd have to tell what color it was based on only green and red, which gets really hard in the blue range. So it would be really hard to tell what color you're looking at in this range here. Or if we didn't have the green cones, we'd have to rely on blue and red. And so it might be pretty easy to mix up these colors in this range. Or if you're missing the red range or the red cones, then you'd have a little bit of trouble telling apart the reds and greens over here. And we can look at a lot of different explanations for color blindness. And as we said, this theory doesn't explain absolutely everything, but it was the first theory that helped us explain color blindness and why some people might have trouble seeing some of the images down here. So in these images, there should be colored dots. So the circles on the outside should be a different color, and numbers on the inside should show up in a slightly different color that should be visible if you have all three kinds of cones. And anyone who is missing some of those cones, one or more of them, could have trouble telling those apart. And I include a little link here. This is just optional. There's a neat website that talks about sort of what stuff would look like if you had different kinds of dichromatism. So if you were insensitive to blue, what would it look like? If you were insensitive to green, what would it look like? Red, so on and so forth. So you can actually go and check out that website and see images that have been adjusted to show what things might look like if you were colorblind. For those of us who are not, it gives us some perspective on how others can see that. Now, as I said, that trichromatic theory seems to explain a good deal of our color vision, and it helps explain color blindness, but it doesn't explain everything. So there was an alternative theory proposed by Edward Hearing in 1878, and his theory is called the opponent process theory. So as we might imagine, they're looking at opposing or sort of contradicting processes that would help us see color. And so here we start off again saying that the human eye has three different types of receptors that have differing sensitivities to light waves. So hearing has agreed that we have 3 different kinds of cones, but they've disagreed on the function of these different kinds of cones. So where this one differs is, instead of saying that each cone responds to either red or green or blue, each cone will respond differentially to 2 different wavelengths. So red or green would be one cone where it would fire in one particular way for red and another particular way for green. There's another kind of cone that would fire for blue or yellow, so have different patterns for blue and different patterns for yellow. And then one type of cone that does black and white, so fire one way for black and one way for white. And this new theory helped explain a couple of things that our trichromatic theory had trouble with. The first of which is simply the presence of yellow. Yellow in our spectrum was actually pretty hard to tell apart, with just having sort of this red and green overlap here. So we have no involvement of blue, and we'd have pretty close responding of red and green, so it was hard to understand how yellow might be well explained using that trichromatic theory. But the opponent process theory tells us yellow has its own particular signal, and that's taken care of in that blue-yellow cone. The other thing that the opponent process theory does a great job of explaining is something called after images. Now, if you went to high school or elementary school in Canada, then you have absolutely seen this image. If not, you've probably seen something similar. And usually it would be in your science textbook. They ask you to stare at the dot in the middle, focus on it very, very carefully. Don't move. Try not to blink. Stare at it for 30 seconds. And then when you're done, look at a blank piece of paper. And what we should see is an after image where we see the opposite colors. So the green shows up as red and the black shows up as white. And we see here, so green shows up as red and black shows up at white. Well, that's showing us that basically those cones that were looking at green, they got tired of seeing green and they end up showing the opposite. So when they're tired of seeing green, they're going to show red. And when the ones that are showing black get tired of showing black, they're going to show white. So we see the opposites. And so the opponent process theory helps explain why, when our eyes have looked at something for a long time and we look away, why do we see the inverse? Why do we see red after seeing green? And why do we see white after seeing black? And the general agreement nowadays, as I've said, neither of these two theories on their own tends to explain everything. So nowadays we use what's called the dual process theory. And this is pretty much a combination of our trichromatic and opponent process theories. So we're going to mix sort of the best of both theories together to have what we think is going on today. So here we have 3 types of cones. We still don't disagree on that. And each type of cone is sensitive to different wavelengths. This is where we're going to get back into the 3 types of cones, blue, green, and red. But now we're going to add in that opponent process part of this. We're going to introduce sort of a new kind of cells, these lateral geniculate nucleus cells, or LGN cells. And these are present in our retina and in our visual cortex, and they respond in opposite ways or opposing ways to red versus green and blue versus yellow. So we add in these opponent cells that would respond to blue versus yellow, red versus green, and also maybe black versus white. There's not a ton of agreement on that point there, which is why you'll see sometimes black and white is included and sometimes not. But what happens to get this blue and yellow is we'd have the blue signal from our blue cones, and the yellow signal comes from a combination or a processed appreciation of both green and red. And then the green and the red opponent cells are going to pull from both green and red there. And then we're able to send distinct signals for blue and yellow and red and green and black, or black and white, all to the brain. So that's our combination of those different theories in order to try and explain how we see color. And this is a simplified version of how we look at this nowadays.